Investment in Caribou Biosciences. It's one of our latest additions. We're actually going to invest next week. We haven't yet invested, but we made the decision to invest. So I want to push this episode out as soon as the decision was made. The purpose of this podcast is to give people a sense for what it feels like to run a hedge fund. And I think this is a perfect example of explaining why we're doing what we're doing. This company fits extremely well into our purpose statement, and we as I said many times on this podcast, we have to align ourselves with our purpose. This is the best strategy to fulfill our mission, which is to generate wealth by investing in fundamentally new concepts and engineering practices with large impact. This is a Not Even Wrong podcast. It's July 31st, 2021. This is Krim Delco. It's a Saturday. There's no update. So for the year, we're up 2%. The market's up 17.5%. We're underperforming by 15.5%. Caribou Biosciences just recently completed their IPO. It's a startup company in the Crispr space. I'm going to explain what they do. I'm then going to try to explain why we're investing in this company, and also related to another company we have in the portfolio, which is Fate Therapeutics. So Caribou Biosciences was founded by Jennifer Doudna and other collaborators of hers. It was, if I'm not wrong, one of the first CRISPR companies, if not the first actual commercial CRISPR company created right after Doudna and her team developed CRISPR in the lab. So what is CRISPR? It's a gene editing technology. It's a very effective, very relatively low-cost, effective, democratizing technology that has proliferated throughout the labs of the world. It allows people to change specific genes in the genome, and therefore opens up doors for therapeutic research, for applications in medicine, agriculture. Anything that life science touches has to do with DNA. Anything that has to do with DNA can be modified with CRISPR, and therefore it fits perfectly fine into our strategy of digitizing life science. This is your sort of writing code and changing the code of life. This is what gene editing is. This is what CRISPR allows you to do efficiently. We've spoken several times before about why we like CRISPR. I just want to summarize two things. One is it's low cost, it's democratizing, and most importantly, it allows try and error at low cost. We've defined this disruption several times on this podcast before. For us at Orange Capital Partners, disruption is defined as lowering the cost of iteration, and this is what CRISPR allows. So fundamentally, therefore, we are highly levered relative to the CRISPR technology. We have several companies in our portfolio, Crispr Therapeutics, Antalya, and Editas, that are specifically working with CRISPR, and then other companies, for example, Twist Bioscience and also PacBio, that are sort of tangentially related to this space. The more research is done, the more the business they will have with their synthetic bio and gene editing, genome reading technologies. So why Caribou Biosciences? Caribou Biosciences, according to their own S1 documentation, is a company that has developed a new CRISPR technology. It's not CRISPR 2.0 exactly, but it's just a more efficient, more efficacy, more lower cost, and allows multiplexing. They call it Chardonnay. It's an acronym for a technology that and I want to keep this high level here because I think it's not necessary for us to go into the weeds. For that, I think we should be talking to the company specifically. Whoever wants to know more details should be talking to Caribou specifically. As far as we're concerned, here's the gist. They've combined DNA and RNA guide to make the few problems that still are very highly prevalent in the gene editing space. One is efficacy, the other is efficiency, cost, ability to multiplex. All these things are still problems. What does efficiency mean? It means that you actually get done what you're trying to get done. Efficacy means that you are able to, once you get done what you're trying to get done. So you want to change the letter A and G in a specific sequence of the genome. You want to make sure that actually happened, and that then you can, let's say, achieve a certain therapeutic effect, and there is no off-target or any kind of bad convolution, lost in translation type of thing happening while you're editing the genome. Multiplexing is the same thing at scale, so you can do many in one shot. Think about sort of parallel programming. Think of what let's say NVIDIA is doing with GPUs. So the ability to do one instruction in multiple areas at the same time. This is kind of what multiplexing is in the gene editing space. I think this is absolutely key to the development of the space, that we need to find ways to do many edits in one shot, so we can at one point do the edits, do the edits, and second, this is very important for research because, like I said in the beginning, lowering the cost of iteration, the faster you can iterate, the faster you can, let's say, take a certain cell and then, let's say, take 1 billion, maybe not that much yet, but let's say take 100,000 cells and then multiplex, change in every one, change one specific gene, but then maybe change another one in the first cell and then another one in the second cell. So you can try and error and see how these changes affect the efficacy and efficiency of gene editing process, and then you have hopefully different therapeutic effects, and then you can faster get to a much better product that eventually does what you want it to do. What Caribou is doing is developing immunotherapy cells, so CAR T and NK cells. Those are two types of cells that the immune system already has. We have that. They attack foreign bodies in our body. And what immunotherapy in cancer is trying to do is use our own immune system to fight cancer cells. What's beautiful about CAR T and NK cells, those are the two types of cells that are being developed in the lab to attack tumor cells, is that they are able to circumvent the problem that our immune system has with cancer, which is our immune system does not recognize the cancer cells, therefore lets them grow, and once they grow, it's too late and we die. So what these cell therapies are trying to do is create cells that do recognize the cancer cells and attack them. So far, we've been relatively pedestrian on this front. There's been a great book I read recently about this. It's about the history of immunotherapy and how it was rejected by the medical community, and then maybe 10 years ago it kind of resurfaced. A very, very important factor of immunotherapy has been the political rejection of the medical community towards this approach. Now, I think we're past that. I think we accept immunotherapy as a therapy, but what we've been doing so far is creating what's called autologous cell therapies. That is, we take the cells from the patient, reprogram them, put them back in, and then hopefully the cells attack the tumor. The problem with that is that often the patient, maybe their immune cells are too weak, maybe they're old already, maybe they're sick already, their immune system is weakened, their CAR T cells or NK cells are not as good. And second, also, we have the immunosystem reaction against those cells. And most importantly, it takes a long time. You have to come in, they take your cells, they do whatever they need to do, and then put them back into your body. What the autologous cell therapy is trying to do, and this is where Caribou is playing, and also Fate Therapeutics, you take off-the-shelf cells from healthy donors, you reprogram those cells so you can inject them in anybody. You deal with the immune system reaction, you deal with the efficacy so they can go attack cancer cells. So what you're really engineering here is two things. You make sure that those cells are not rejected by the immune system, and you make sure that they actually go kill the tumor. These are the two key things that need to be engineered. And that's what Caribou is trying to do with this new technology. That's what Fate Therapeutics is doing also. And now I'm sort of jumping back and forth because these are the two companies in our portfolio that are in the business specifically of creating off-the-shelf cells to fight cancer. Why are we invested in this? We've said this many times, fundamentally new concepts and engineering practices with large impact. Now, these are fundamentally new concepts. it is, in case of Fate, they're taking iSPCs. So these are cells that are turned into stem cells and then reprogrammed back into becoming NK cells, killer cells, to go after tumors. Caribou is sort of a next level here because they're using advanced CRISPR technology to do the same thing. When it comes to Fate, there is some CRISPR technology being used there. It's not clear to us what exactly they're using, so I don't want to be too harsh on them, but it seems to me that Caribou is probably the next level here in terms of applying more advanced CRISPR technology, which has better promises for success when it comes to programming cells to do what they need to do. What we're doing here is programming cells. So we're engineering cells to do something specific, in this case, attack cancer. And this is why this fits perfectly into our portfolio. So we have a fundamental new concept, we have new engineering practice. This is a manufacturing problem, this is an engineering problem, this is of course also a science problem. It's not 100% clear what exactly needs to be done. So there's a lot of research still being done. There's a lot of try and error on that front being done. So we're absolutely in new territories here, and therefore we have to acknowledge also the risks of this approach. There is certainly a lot of technology risk. There is, can this be even done type of risk. I think the upside is humongous. We've seen that already with some of our portfolio companies, namely Intellia. When things work out, Intellia has almost 10x since we invested in the company. So that's it. That's the risk reward here. I think there's not much more to say here in terms of when you invest in these types of technologies where there's a lot of technology risk, there's a lot of even scientific risk still, but given the success of the CRISPR technology as a fundamental platform technology to use it to engineer cells, given the success of the last decade, we are fairly confident that the technology platform itself has a lot of legs. Caribou, given the scientific talent there, is certainly one of the best positioned companies to take advantage of this. And then the actual purpose of the company, which is to generate off-the-shelf cancer-fighting cells for patients, is just unbelievably interesting for us, because it is exactly along the lines of lowering the cost, increasing efficacy, making lives better for people at lower cost, solving fundamental problems such as cancer, lowering the cost of doing that. It couldn't be more aligned with our strategy. So to summarize, we are going to invest in Caribou Biosciences. I think initially we're going to take about a 4% position. We are going to invest in Caribou because they are advanced in the CRISPR space and using that technology in specific areas of cancer immunotherapy, which is another area that we are very excited about, because we believe that this is exactly the kind of technology that we should be trying to implement in cancer fighting research, because using our own immune system to fight cancer is ultimately the best way. We kind of piggyback on natural selection and evolution. We have a very effective immune system. It just doesn't work very well with cancer, so maybe we can re-engineer some cells in our body to actually do that. We do that off the shelf, so in allogenic cell therapy, which is, we can do it off the shelf at low cost. We can take healthy cells from healthy donors and inject them in sick people, in particular older people that probably already have a weakened immune system. They can take advantage of this, or in general, let's say if you have AIDS or any of that stuff, your immune system's already weak, and then you, God forbid, you get cancer, this allogenic cell therapy can be a real savior. And Caribou has developed this Chardonnay technology, which is a combination of RNA and DNA guide. The guide in CRISPR technology is a sequence that is used to, on the one hand, define where specifically a Cas protein has to bind, and it also binds the Cas protein. So the guide is a synthetic sequence, a molecular sequence, which is used for direction and binding of the Cas. This is where the actual changing of the genome happens, when you define It started with RNA guides. Now the Chardonnay technology is a mix of DNA and RNA guide. So think of this as a molecular sequence which is used to go to a specific place in the genome and then bind a specific Cas protein, which then does the erases a certain piece of the genome, and then we're using our own cell recombination technology to rebuild the genome, or we fake it a little bit to change the genome to sort of write instead of A, G or T, C, and so on and so forth. Thank you.